detection
sensitive,
is
yeah.
super.
Right.
Are you really worried about like mass separation?
I'm not as doing like isotopes.
Okay.
Isotopes, you'd have to do something else, but it's more just establishing that a metal is present.
Gotcha.
All right, everyone. Thanks for coming. No rush. Take your time getting food and everything. And as we start, we're also going to be streaming this to YouTube. So if you have any friends who couldn't make it, just let them know that the Georgia Tech Astrobiology channel is streaming this, and it will also be available recorded afterwards. So yeah, whenever you get a chance to get your food and take a seat, please do so. So first, I'll just start by mentioning Georgia Tech Astrobiology Fellows who organized this. There's 6 of us who were awarded the Georgia Tech Astrobiology Fellowship this year, and we organize a lot of astrobiology events and talks throughout the year. So this is our kickoff event today, and it's starting with this talk by Dr. Aponte, and then later today we'll have a social at 4 o'clock, and that'll take place at the Mosey patio. So that's just over there to the north of us, down the steps of the BioQuad. And so that'll take place at 4 o'clock. And we also have an event in the spring, which is our basically a mini conference, which takes place with all astrobiology and space-related researchers in Atlanta and the Southeast. And so we welcome you to come to that and to be on the lookout for more information. So,
thank you.
Dr. José Aponte is coming to us from NASA Goddard Space Flight Center from the Analytical Astrobiology Lab. And Dr. Aponte did his PhD at University of Louisville and did a postdoc at Brown, and then joined the AAL and has been working there ever since. And so we're really excited to have him talk today about prebiotic chemistry, the origins of life, and sample return missions. So I'll let him take it away.
Great, thank you, Chad. Can you hear me well? Okay, so yeah, I work at the NASA Goddard Space Flight Center Astrobiology Analytical Laboratory with this group of fabulous people. One of them, previous graduate student from Georgia Tech, Eric Parker. So you may remember him. Or maybe not, I don't know. So there are different ways in which we can study prebiotic chemistry. So I'm an astrochemist, and when I say that, some people tell me, like, you know, are you an astronomer? And I'm like, not really, but that's a good way to do that, right? So we have satellites in outer space, satellites on the Earth that can detect different signatures or different molecules that are hanging out outside the Earth, right? And that's a good way to know what's the inventory of material that it's in locations that were similar to our solar system before the Earth and life, you know, happened. But there is a different way in which things can be done, right? So interstellar material, or say extraterrestrial material, can be measured in situ, right? So you send a probe, and you land in a body like a comet, like, for example, the Rosetta mission did, and can detect molecules of life, for example, amino acids. All right? And this is one of those detections in which Rosetta detects glycine and some other smaller molecules that could make glycine, which could make life eventually. However, in our case, we are, or at least I am, an experimentalist. That means that I work in the laboratory most of the time. And I do a lot of analytical analysis, right, using different instrumentations. We could, for example, mimic what happens in between galaxies, or we could mimic what happened during the formation of a solar system, or even we could mimic what happens after the Earth finally formed and what sort of chemical processes led to the origins of life. But as much as we can mimic stuff, a lot of the chemistry that we could also analyze already happen in the solar system, right? And if you think about that, we have asteroids, we have comets, and those are relics or remnants of everything that didn't become a planet or didn't become a moon, right? And all that chemistry that we mimic in the laboratory has already happened in asteroids, for example. And meteorites, which are fragments of these asteroids that land on the Earth, contain all that chemistry that we can analyze and process in the laboratory. In this case, for example, we will look for molecules of life like amino acids, and we can determine their molecular diversity, their abundance, and so on, using different analytical techniques. And why would we do that, right? Why there is a need to study meteorites? It's because the Earth is completely filthy and contaminated with biology, this disgusting biology that you all know, and geological processes, right? They have deleted a lot of the evidence of how life happened on this planet, right? So we need to look outside. We need to look to samples that have not been on contact with this biosphere. And again, those are meteorites. Meteorites can actually fall anywhere on this planet. Of course, the Earth is covered with water, so the majority of the meteorites will land on the water in the ocean, right? But they could also land in locations like it happened in 2019 in Costa Rica. This is a place called Aguasarcas. And there was a meteorite called now known as the Aguasarcas meteorite, which almost hit poor Rocky here. Rocky's fine, but his home is not. And anyways, they can fall everywhere. And if it happens to fall in your backyard, you can actually make a lot of money out of it, right? You can sell it online. But because I know you are decent people and decent scientists, will donate it to a museum, right, or a laboratory like mine to do analysis, of course. Samples like these are not that useful because they are like contaminated with these, hands. So in the event that you happen to be rocky, and a meteorite falls in your backyard, just use gloves to collect the sample. Collect a little bit of the dirt that is around the sample so we can compare and freeze it, right? But not next to your Chinese food because that would also contaminate it. Anyways, in my lab, what we do is we try to get cleaner samples. Those cleaner samples will come from Antarctica. Why? Well, because Antarctica is a dry place. It's a white place, which means that rocks outstand. And so NASA and the NSF send people, that program is working back again after the pandemic. They send a group of people to enjoy a really warm Antarctic summer for 3 months. So those days that they are not running crazy, having deep thoughts about their life, they go on the ground. and there's a nice book about that, by the way, by my colleague, Danny Glavin, which is true. Isolation makes you, COVID-like symptoms. So they go on the field and they collect those rocks, and they bring them to the Johnson Space Center where they get cataloged. They are analyzed and they are put into different types of meteorites, classified. And so that's how we get cleanish material, right? This is the best we can afford on this planet. And also Antarctica has the ice sheet is actually moving all the time. So it kind of, if a meteorite landed, say in one spot, a year later will be buried. And maybe, 5 years, 10 years later, it will show up somewhere else. So that's how it's a never-ending place where you can get meteorites, okay? Meteorites are classified into 3 main families, but the ones that we care the most are the stony, right? Not the metal ones, because, you cannot really, it's hard to find organic molecules into molten metal, right? And in this type of stony meteorites there are two main classes, which are the chondrites and the achondrites. And the chondrite is just a fancy word for a really old inclusion. So these white little inclusions that you see in this sample are the first solids of the solar system. They are older than the Earth. They are even older than the sun. Because when the sun was finally forming, these calcium aluminum inclusions, or chondrites, or sorry, chondrules, were already formed on the backside of the solar system. And they were accreted, included on this dust, on these bodies that were not affected by planet formation and so on. So they have experienced all this formation of a solar system. They have survived it, right? And therefore, they have encapsulated all the inventory of the chemicals available during the formation of our solar system. Among the chondrites, we have the carbonaceous, which is another fancy word to say that they are rich in carbon. And the carbonaceous family, so if you combine those two, we study carbonaceous chondrites, are separated in a subset of different subfamilies that are aligned according to their level of water or thermal alteration. It means that some of the meteorites contain a lot of water, some of the meteorites have experienced high temperatures, right? And somewhere in between. So the classification is actually kind of blurry because there are meteorites that contain a lot of water and have experienced high temperatures, and there are meteorites that do not contain water but only have experienced high temperatures, and so on, right? And so based on those conditions, we can get the sample, process it, analyze it, and look for organic matter. Why organic matter? Because we are made of organic matter, right? And if you think about it, those molecules are actually found in these meteorites. How many molecules? Millions of them. So there are two specific ways in which we can target or we can approach this problem. We can go ahead and try to name each of those million individual species that are present, or we can target the analysis of specific families of compounds. The families that we care about are the ones that are important for life. That includes amino acids, carboxylic acids, which makes this cell wall. Amino acids makes the proteins. Nucleobases makes DNA. And some other compounds are analogous or structurally related to amino acids, for example, so that they help us understand their synthetic origins. So this is, of course, in the context of the origins of life. And as you know, there was a period of time in which the Earth was a little bit hot, and there was a lot of impacts, which brought some of the water, some of the organic material that we could have used, well, not we, but I guess nature could have used to develop life, right? And although meteorites and microparticles, cosmic dust, still falling on the Earth, during that time it was a lot higher. So that's one of the links between origins of life and the study of meteorites. The way in which we approach this question in our laboratory is we study a specific family of organics, say, for example, amino acids in carbonaceous chondrites. And by looking at their isotopic values, molecular distribution, and chirality, is that we can assess how the solar system formed, what happened in the asteroid, and how it helped to the origins of life. And if you haven't seen these words before, don't worry. I'm going to explain you quickly what exactly is the isotopic values, molecular distributions, and chirality. But I just want you to know, like, this is the reason we do it, right? By analyzing the molecules in the carbonaceous chondrites, we can not only understand how was the molecular inventory when the Earth didn't have life, but also how the solar system evolved. This is our laboratory, NASA Goddard. we are fully and really well equipped. So if you are in the DC area sometime, stop by and visit. We have good setup with a bunch of different instruments that allowed us to not only identify the structure of the molecule, but also determine their abundance, and also assess if the molecule indeed is from outer space or terrestrial, meaning terrestrial contamination. I mostly do gas chromatography, and we have a two-dimensional gas chromatography instrument that maps the entire mix of compounds in the sample. We have a hybrid GCMS, IRMS that gives you the nature of the compound, if it's terrestrial or extraterrestrial. And we have some high-resolution HPLCs, or liquid chromatography instruments, connected to orbit traps and high-resolution mass spec. So yeah, we're doing good so far. I mean, those are really expensive instruments. So this is what happens to a meteorite in our lab. So we get the sample, and it gets powderized. We make a powder out of it, which then it's extracted, which means that the powder is in contact with a solvent that extracts specific families of compounds, right? If you want to look for non-polar compounds or something like gasoline that doesn't mix with water, then you will use a non-polar solvent like dichloromethane. But if you want to use amino acids which don't dissolve in non-polar solvents, you use water. And so that's how you get the compounds out, do a chemical cleanup, and then a chemical derivatization, and do the analysis, right? In this case, for example, this is an HPLC trace of different amino acids. OK, so this is where it gets a little bit tricky. We do study isotopes. Again, that's important because we want to know if the molecules that we look at are from the Earth or if they are extraterrestrial. To do that, we use an instrument that is actually developed for the pharmaceutical industry. And so have you ever thought about how in the Olympic Games they know if someone is cheating or not? Right? If someone sniffs cocaine, it's really easy. You get a sample and look for a cocaine derivative. It's present or not. Clean, right? But what about if someone gets injected with microdose of testosterone, for months? I'm not giving you tips, but anyway, so testosterone is made in your body. Like women and men both produce testosterone. And so this instrument looks at the amount of carbon atoms that are generated that are in this testosterone sample, for example, by your body. So what you make in your body is out of the diet that you have, right? So that's why, like, imagine we all were eating the same thing for a month, and they take a sample, we all will have the same kind of atomic isotopic value because we are all eating the same thing. But what about if, like, Jose was injecting some testosterone made in Shanghai, right? Out of like ammonia from Singapore and formaldehyde from Mexico, or I don't know, right? So that will have a different carbon content. And so the isotopic value of Jose will be different than the average. And that's how they are detected. In the same way, terrestrial and extraterrestrial samples can be differentiated because biological processes use the lighter atom, which is carbon-12 and hydrogen, to make biomolecules. Whereas in space, biology didn't happen, so the amount of carbon, lighter carbon that is incorporated into chemicals, it's lower, so they contain a higher ratio of the heavier atom, which is carbon-13, and that's why we use the isotopic ratios, right? So the molecules that are from outer space contain the heavier isotopes, and the molecules that are from the Earth contain the lower isotopes, and that's pretty much it, right? So that's how we detect terrestrial versus extraterrestrial. We also look at the molecular distribution what do I mean by that? So there are several types of amino acids. There are the alpha, beta, gamma, and delta types. And all of them like, have a different branching structure, right? Biology uses the alpha type. So this one's in blue here, right? And if we look at the molecular distribution of meteorites, we can see which amino acids or which type of amino acid it's made is present in the higher abundance. In this case, we see that, for example, the alpha type are more abundant, and therefore that would suggest that they were made through a specific type of synthetic process called the Strecker synthesis. It's not the only chemical pathway, but it's probably the most reliable or more the main one, right? So, and if we do the same thing and we analyze a bunch of different meteorites that contain a high amount of water or have experienced high temperatures, we see that they have different ratios of different types of isomers, the alpha, beta, gamma, and delta. So if I would ask you a question like, if I give you all the money in the world you have, or we have, to build a mission, a space mission, and I tell you, I want you to go to an asteroid that has amino acids like the ones that we have in our body. What asteroid would you go? Would you go to a thermally altered? Would you go to a aqueously altered? Or you would go through something that looks like this that is rich in alpha amino acids? Probably go there, right? And so that molecular information, it's important to that level, okay? And we finally, the third thing was we look for the chirality, right? So chirality is actually a very simple thing. It's like, atoms can be similar but three-dimensionally opposite, right? And so we have amino acids that are left-handed, and we have amino acids that are right-handed. And biology, it's made exclusively out of the left-handed only amino acids. And we honestly don't know why. We don't know why they decided, nature decided to use one over the other. The thing that we know is that we are made of left-handed. And that all of the amino acids that, well, not all, but 19 of them out of the 20 that are used in biology are the alpha type, right? So those are the two clues that we have. We have they are alpha, similar to those that you can find in meteorites, and they are left-handed. Would also be similar to the ones that we have in the meteorites? Let's see. So to understand this, to answer this question, we look at a specific molecule that is called isovalene. Isovalene is an amino acid that contains an amino group, an acid group, but also a methyl or an R change, alkyl change in the alpha carbon, right? So this molecule doesn't dissociate, meaning doesn't racemize, doesn't break apart. So if it's born left-handed, it's going to die left-handed. If it's born right-handed, it's going to die right-handed. Okay? So you cannot really alter it through processing. And we have found that this isovalene, this amino acid, which is also an alpha amino acid, contains high enantiomeric excess. What does it mean? That left-handed amounts, or the left-handed versions of the amino acids are present in higher abundances than the right-handed amino acids, just like us, right? So, and to understand why isovalene shows that pattern, we look at other molecules, like, for example, amines, which are structurally related. So instead of having an acid group, we have a proton or a hydrogen there. So these molecules contain the same number of carbons, except for the carboxylic acid. But they share the same aliphatic chain there. And we can expand this story to other families of compounds. So because amino acids are typically made of aldehydes and ketones, and they will, for example, isovalene will be made out of the aldehydes or ketone, and that could become an amine through decarboxylation, or an alcohol through reduction. It could also be that the starting material for the amino acid would become a hydroxy acid, and then it could become an aldehyde or even a different amino acid, right? So all of these families of compounds are interrelated, right? And so in my lab, when I got there in 2012, we were looking only at this one. And so now we can look at other different families. And so now we can understand their abiotic origins and their chemical relationships, right? So, and coming back to the enantiomeric X story, this is how isovalene perform in the meteorites that we have studied. And we see that it shows some large enantiomeric excesses in the meteorites that contain high levels of hydration, high levels of water. Okay, so we have a relationship there. Meteorites impacting the Earth, bringing water, bringing organics, bringing amino acids that are enriched on left-handed amino acids to give birth to life, which is also left-handed. So there you go. There you have your theory, right? So the impact of these extraterrestrial bodies brought an excess of left-handed amino acids, which later kicked the audience of life, influenced the audience of life to be left-handed, right? And we see that by analyzing the amino acids and analyzing the structurally related amines, for example, where we've also done the carboxylic acids, and they both show the same, in which the amino acid is left-handed enriched, and the structurally related molecule like the amine or the carboxylic acid is not. And why is that? Well, we have to then think of how is the amino acid made in outer space, right? So imagine the cloud before the solar system was formed, right? Having only ice and dust and some radiation going through, in which this ice will deposit certain atoms. The atoms start depositing and evolving with this radiation, and there is where you start making more complex molecules like amino acids. This process has been mimicked in the lab, and it happens that if you irradiate or give a sort of type of UV light that is left-handed, you will make left-handed amino acids. If you do a right-handed version of the radiation, you make an excess of the right-handed version. Okay, so that is called the breaking symmetry. So from the get-go in the cloud, you are breaking symmetry, and then there has to be an amplification process. The amplification process will happen in the parent body through the aqueous alteration. So bear with me. We make amino acids that are enriched in the cloud. We make a protoplanetary disk. We make the bodies, which encapsulates the water that process the meteorite. And this water will do perhaps an amplification process in which the solubility plays a huge role. And that's how we not only break the symmetry, but amplify the initial chiral excess, seed on the Earth, make life that is left-handed, right? And we know that amino acids can easily be broken, can break symmetry when you do that type of chemistry, but other molecules like the amines and carboxylic acids don't, because they don't absorb the polarized light, the ultraviolet light, the UV light, in the same way. So everything, falls into place, right? We use radiation, we break symmetry, we amplify, and so on. So let's quickly rewind everything. We can look for organic compounds in meteorites. We can find a connection between the audience of homochirality that make us, and we can study that by analyzing different families of organic compounds, right? Everything makes perfect sense.
right? So we don't have to do any more work. The origins of life is solved, right? Yeah, no. It's not because there are a lot of holes in our story, okay? So, for example, we don't know the inventory of these small molecules that eventually make amino acids, for example. So if you think of that, if the amino acid is coming from the aldehyde, where is the aldehyde coming from, right? The aldehyde is coming from carbon monoxide. And, okay, I'm not going to go like where that carbon monoxide comes from, right? But we don't know how much of that was present when our nebula, when our protoplanetary disk was formed. In the same way, we don't know any other of the starting or potential starting materials of amino acids. On top of that, the isotopic values that we study that are, say from the time that we make this asteroid, maybe have evolved because those processes that happen in the asteroid may have destroyed stuff and may have made stuff. That's called fractionation. We don't know how much is made, how much is destroyed. And so the carbon atoms that we see in each amino acid is random, right? We don't know exactly where it comes from or what is the starting material for each of them. And as you can see, there is a huge void into the inventory of the starting materials. To make this more clear, it's like you have two molecules. They contain different levels we will have a pool of carbon atoms, could be carbon 12, carbon 13. And this is what we see, in the analysis, but we don't know where they came from. Like in our analysis, we are not accounting for the original budget of those molecules because we don't know them, right? And so, so there are two main challenges, which are two main opportunities for graduate students like you. You can base your entire career on this. The first one is that we don't know the abundance, right? We don't know how much of the starting materials was present when the asteroid was made. So we are only analyzing what we see today, but that doesn't necessarily mean that what we see today was the same as 3-4 billion years ago, right? And so that fractionation is unknown. That's something that needs to be determined. And so I propose that we need to do a synergistic approach. We need to, ask theoreticians, doing people computational chemistry, doing experimental work, observational work, but even with that, we'll be kind of always in the limbo because our solar system is already formed. Right? It's gone, right? Like we don't know what was the molecular budget when it first formed. So all the indications that we see from locations where we look at outside the Earth, they may not be necessarily the same as ours, right? So how do we overcome that thing that nobody thinks at, right? Nobody, like when you as an astronomer, but yeah, how do you know that that's what happened to us? I mean, you assume that it's the same, but you don't know, right? Anyways, and of course, we need to think quantitatively, right? That's something that we're not doing so far. We can measure the compounds that we have in storage in the samples, but how the isotope effect, the isomeric effect, and all the nature of the compounds has been impacted by the fractionation is also something that nobody talks about, and which is fair because we are, again, this is a huge opportunity, right? Like we have only started to develop methods to see the molecules. So now that we can see them, what do we do? What's the next step? Have quantitation and, for example, calculate the stability and stuff like that. So So yeah, it's kind of bad news, but also good news. So because I work at NASA, I don't have the chance to only be happy analyzing meteorites. I have to think about the next space mission that is going to be the greatest thing on Earth, right? And the greatest thing on Earth that we are working on right now, it's sample return missions. Because as much as meteorites are great and exciting, they have been exposed to the terrestrial biosphere, right? And they may or may have not been contaminated, right? They have been altered by crossing our atmosphere. And so that's when we think about, okay, why don't we go to the source and bring it to the lab, like instead of waiting for Rocky to don't chew on the meteorite. So the first type of sample return missions, it's the Apollo missions, of course. We're very familiar with them. They brought like tons, but several kilograms of materials to us. Stardust brought grains of a comet, and now OSIRIS-REx. Just bear with me. So OSIRIS-REx is actually an acronym that stands for Origins. Origins is origins of the solar system and the origins of life. Spectral interpretation is because we mapped the surface of Bennu, or the asteroid that we went to, very well, and we know its composition, its surface. We do resource identification because this is not sci-fi, but eventually we want to do an asteroid hop, right? Getting the water in the asteroid as a fuel, and right? Okay. Well, it's sold, so it's all right. Security, because there was a tiny chance that Bennu could impact the Earth in the next 200 years. So we want to understand its orbit and see how it changed and what's the probability that it will damage this planet. But the most important part for me is this here, is the regolith explorer, which means that we are going to be bringing a piece of the asteroid to the Earth this Sunday. Yeah, so why do we pick Bennu? Bennu was picked out of the immense amount of asteroids that we have in our solar system for a few important reasons. It was near Earth, which means that we can complete the work in a lifetime before I retire or before, it had a good orbit because it was between the Earth and Mars. So it has the right size. It is not too big, not too small. If it's too small, it's been too fast, and then we could not approach safely. It could be damaged. If it's too big, it will be too heterogeneous and not representative. So, but most importantly, we pick Bennu because it's a carbonaceous type chondrite. It's a type of asteroid from which we think we've gotten meteorites on the Earth, so we can analyze all the meteorites and understand Bennu, right? We have a lot of information on how the meteorites look like, and therefore we can do the same thing with Bennu. And so back when the mission was picked, this was the best image that we had of Bennu, and this is what we know now. So in the previous figures, you could see that Bennu seemed to be a very smooth surface, very planar, flat, right? Really nice to land and touch. But when we got there, it was like, what is this? It's so rough, so many boulders, and where do we land? Where do we land safely, right? On top of that, stuff was coming off, can get heat. We are getting an asteroid shower in November from Bennu. So that was the main challenge. When we got there, it was completely different to what we expect, right? What we planned for. And luckily, we were able to orbit for 2 years to map the whole surface, understand really well where can we land. And on October of 2020, that's what we did. So we had this robotic arm that was equipped with a sort of a car filter connected to a nitrogen tank that was going to be flashing gas or nitrogen gas so that the rocks and pebbles would be lifted from the surface, because again, we thought that the surface was going to be flat, and so those materials could be collected, trapped in the capsule, trapped in the filter, and but in reality, the surface of Bennu was a lot more complex and rugged than we knew. The mission is going to be successful if we bring as much as 60 grams of material, but we can collect up to 2 kilograms of material, which is called the regolith, or surface of Bennu. We know Bennu, it's very similar to carbonaceous chondrites, specifically those that are aqueously altered. And that's phenomenal because, as I showed you before, those that contain high levels of water also contain the highest levels of enantiomeric excess of left-handed versions of amino acids. So we hoped that we can find large enantiomeric excesses of left-handed amino acids, and that would be huge for knowing why we are homochiral, why are we made of left-handed amino acids, right? So if that happens, and that's what we find, it will be a huge deal for humanity. Yeah. So again, Bennu looks like aqueously altered meteorites, but it doesn't truly fit the line of any of the meteorites that we have in our catalog. So there will be a lot of work to be done trying to assess which of the 40,000 meteorites that we have in the catalog match the best to Bennu, right? And we have not analyzed 40,000 meteorites, we have analyzed like 50, 60, right? So there's a lot of work to be done. On October 20, 2020, we approached the surface of Bennu, and this is the real footage of it. We touch it, and this was the second surprise that we had from Bennu. So the whole tag and go or tagging sequence was controlled by computers and artificial intelligence, and there was an 8-minute delay from Bennu to the Earth. So we didn't know what happened, but the instrument was equipped with sensors that could abort the mission if they found any problem with it. The second surprise that Bennu gave us was that it was way softer than we expected. We were expecting a lot of resistance from the surface, and that's why we had those nitrogen tanks to blow stuff, because we were able to lift stuff from it. But as you can see, we went deep up to 1 meter through the surface of Bennu. We were this close to lose the robotic arm, actually. Yeah, so, but the good thing about it was that we were able to collect so much material that the capsule or the filter was completely full, and actually we were losing material, over some time. So the spacecraft had a system in which it could rotate on its axis and measure the speed, change the speed of rotation so that it can quantify how much material was collected. But we decided to avoid that so that we save as much material as possible. So we don't really know how much material we're going to be collecting, but we know that it's going to be a lot more than 60 grams, or at least that's what we hope. So from that angle, we're gonna do good.
Navigation.
This is the day of the
Program manager.
Program manager is go for tag.
election for that touch and go maneuver.
All stations, this is OREX Systems on OREX Chord. I copy all subsystems or go for tag.
Position uncertainty is 0.5 meters. Predicted tag lateral offset is 1.7 meters. Hazard probability is 0%.
There is actual footage. You can see those white spots. Those could be carbonates, which means a lot of water, which means a lot of alteration, and those hydrothermal processing, right? So that could be really.
OREX has descended below the 5-meter mark. The hazard map is go for
We don't have
tag.
any control over this.
And we have
So we were definitely excited about it. That's the lowest the mission. And yeah, so that's us departing Bennu and coming back to Earth. And so we have several cameras in the system. This is what I told you, like we were losing material from it. So we decided to encapsulate it, don't try to spin it. So that if we spin it, you lose more material. So we just encapsulate it. This is the crater that we left in Bennu. You can see the thrusters marks. And yeah, so actually that's the next image that I had. So this is how it looked before we touched the surface. And this is after we touched the surface. You can see the crater that we made in the surface of Bennu and the craters that the thrusters made. So the thrusters were pointing out, so we don't think we're contaminating the samples. And besides, we went so deep inside the asteroid that should be safe. And on this Sunday, the capsule is going to be dropped by the spacecraft, which in turn will continue its pathway to a different asteroid called Apophis. And the OSIRIS-REx mission will change name to OSIRIS-APEX mission. So we're going to a different asteroid. We're not going to attack that asteroid, but we're going to orbit and just evaluate it. And so the capsule containing the material is going to fall in the Utah desert in this military range where stardust also fell. And I mean, we know the terrain, weather is good, so things should be okay. We've been practicing for this a lot. We've mimicked everything that will happen that day. We will be moving, collecting the capsule, bringing it to, so we're gonna be collecting samples from the background. We're gonna secure the sample and so on. And it's gonna be put into like an inert atmosphere of nitrogen gas, moved into Johnson Space Center following day, where it will be opened up. We're gonna do a quick observation. And if the government doesn't closes, we should have information for you guys the following week. It's really exciting. We've been practicing a lot. We've gone through everything that we will be doing with the sample with mock material. So we have the same people doing the same experiments, and everything is really well constrained. Nothing is left, out of. The group of people working on OSIRIS-REx is pretty diverse from different countries and even continents. People at Goddard, that's where I am, will be doing the very initial analysis. So we're going to be doing what is called the preliminary examination, the PE. So we're going to be the first ones getting samples from OSIRIS-REx. I think I'm going to be the first one taking a look at it in the lab. So it's really exciting. We've been working for this on so many years that now I just want to get done with it. So it's fine. But OSIRIS-REx is a mission that is going to keep on giving, right? Because 75% of the material will be safe for future generations. So you'll be able to request your own sample and do analysis. And even your kids or, future generations of not only people but instruments will get beneficiary from this mission. These are the milestones that we will try to assess. We'll get their results maybe by October 11. And so that's where we're gonna try to provide first what's the total mass, how does it look, a quick look of how it is so far. Then we'll be cataloging all the material that is collected, and so you'll be able to request materials. And we're going to have a bunch of papers being published through the next 2 years, and that's pretty much it. Thank you very much for having me here. It's been an honor, and join us on Sunday to celebrate OSIRIS-REx. And so, yeah.
All right. Thank you guys so much. Thank you, Jose. This was a fantastic talk. I learned a ton of things, and that's even coming from someone who used to work with Jose in the AAL. So I'm sure plenty of you have questions, and we also have questions online, I'm sure. So I'll come around with the microphone, and first person to raise their hand wins.
Yeah, Jose, this is great. I was just wondering, is this actually the deepest extraterrestrial sample that we've gotten back so far?
The deepest, yeah, for sure, right? We've worked with Hayabusa2 materials, but they are really surface. Actually, Hayabusa, it was really nice because it brought two different types of samples. So one sample that was collected just from the surface of the asteroid, but also a sample that, so they use a bullet, like a ball, that they shoot into the surface, create a crater, and then collect that material. So they have two different types of samples. They kind of look the same, honestly, but they are not deeper than maybe I don't know, 5 centimeters. So
Well, I think even Apollo only got maybe a meter deep.
Well, yes. So those I didn't mention this, but we also analyze Apollo samples as part of the ANSA.
Yeah.
So and we analyze this core.
Yeah.
So the astronauts were kind of crazy back then. Like they not only stand up in the moon, but they also core it, make throw a tube to the ground, and they close it there. They seal it in the moon and brought it back to the Earth. 60 years later, we decided to cut it through, open it up, and we analyzed that material. I mean, we didn't find, we just submitted a paper on this, and the organic material that we looked for in the moon samples. So you will know more of what we found. We found some species like carboxylic acids and cyanide, but that's pretty much it. But other than the moon samples, yes, these are kind of the deepest non-radiated materials that we have. Yeah.
Did you do organic analysis on the Ryugu samples?
I did.
Yeah.
I did. So we were part of the Hayabusa2 soluble organic analysis team, and we did amino acids, amines, carboxylic acids, pHs, gases, and nucleobases and sugars. So the concentrations are published. It's kind of variable, but all of them pretty low. Okay. You can request Hayabusa2 material now. I got 10 milligrams of Hayabusa2 to do cyanide analysis, but didn't get any cyanide out of it. So yeah. So yeah.
Thanks so much for a wonderful talk. It's so exciting to have you here 2 days before OSIRIS-REx return. So I was just curious logistically, once you're done doing extractions and analysis in your lab, what's the long-term plan for those samples? Like, are they able to be analyzed in other methods after you've done the techniques necessary to analyze them?
OSIRIS-REx, nothing has been left to chance, right? Every single grain of OSIRIS-REx has to be accountable. And we have a software that monitors and tracks the sample itself. So once you receive a sample, you have to tag it, create a log for it, and then everything that happens to that single grain or dust, it goes into a catalog forever. That catalog will be publicly available, so you will know when it left JSC, who used it next, who did, every single step into this app. It's kind of annoying actually because I have to do my analysis and be like writing my page, writing my notebook, do the analysis and tag everything, writing in the app, like, I put it into water, I heat it up, I did this. And if you have several samples, it's just, but it's for the greater good, I guess. And yes, those samples will be available later. Yes, that's what's happening.
Yeah, thanks so much for the talk. As a computational chemist, I know you mentioned integrating something along the lines of simulations into your workflow in the future. I know that computational chemistry especially plays a role in like the detection of rotational and IR spectra and like the interstellar medium, but I'm curious, like, could you elaborate a little bit more on what you're hoping to do with computational chemistry in your workflow?
Yeah, so there's several things that could be done, right? I would like people to model, well, from what we see now in the inventory of organic molecules, what was before, right? So if we tell the computer, the software, or the algorithm, this asteroid contains this much water and have seen this as the highest temperature possible. And contains these amounts of like different species. Can you tell me 3 billion years before, what was the composition of it, right? And if so, can that be similar to other observations that people have done of other protosolar places, right? Which one is the more equivalent to ours, right? So those, I mean, probably it's science fiction, but I don't know. I don't know anything about computers. But we can only tell you what is there now, and I want to know if that's true. How accurate is that to what was before, and that will also inform us on what are the conditions that we could like mimic in the laboratory to do more processing, and so now we can do the opposite, right? We're going to start from the scratch, starting materials, and evolve, right, from like carbon dioxide or carbon monoxide to isovalene to proteins, right? So it's yeah.
All right, we're going to go to one of the online questions.
All right, one of our first questions online is, why is isovalene resistant to racemization, and can this be explained by chemistry? Can it be explained by chemistry, like the racemization?
So, yes, so isovalene, it's an alpha alkyl amino acid, and that's the answer. So it's just that it's not that it's not racemizable. It will racemize, but at slow pace, it will take a lot lower than the 3 billion years of existence to racemize isovalene, right? So that's why it's negligible. But essentially, I mean, of course, if you radiate it with UV light or, I don't know, like gamma rays, of course you're gonna break it apart, you're gonna dissociate it. But if you just put it in water and heat it up for a billion years, nothing's gonna happen to it, right? So, it's because it's an alpha alkyl amino acid, and some other alpha alkyl amino acids are also hard to racemize. That's yeah.
Another one we have online is the isotope signature of a carbonaceous chondrite distinct enough that the stable isotope analysis could show that bacteria had grown on C from that one?
Yes. Yes, if bacteria had grown in the carbonaceous chondrite, you will see its isotopic value being completely different. And that's exactly how we know that contamination was present or not. Yeah.
If there's another mission like OSIRIS-REx, what target would you like to pick and why?
I don't know the catalog of asteroids. That's but definitely going to a aqueously altered carbonaceous chondrite would be nice to have more data points. I mean, hopefully we can get enough exciting data out of OREX to not only go to another carbonaceous chondrite type asteroid, but maybe a comet, or maybe somewhere like a Jupiter moon or Saturn moon. We have MSR also that may happen. So, so there are a lot of different exciting places in the solar system that we could go to. And of course, the deeper you go into the ground, the better chances to get more information about.
And for those who don't know the acronym, MSR is Mars Sample Return.
Oh, yes.
Okay. Thank you, Jose, for a beautiful talk, really inspirational, and I learned a lot too. Could you tell us what, to the extent that you're allowed or want to, what do you expect to find in the OSIRIS-REx samples, and then what would be something that you're kind of really hoping to find above and beyond?
Absolutely. So this is public information, so you can look for it, you'll find it. And I was expecting this question. It was like, I didn't talk to you, but I knew someone was going to ask about that, so I came prepared. So OSIRIS-REx and at least the soluble organic analysis team, well, actually, the OSIRIS-REx mission, because we cannot simply say, oh, we're going to answer so many questions, that's great. No, we have to make a list of questions that we need to answer. And to do that, we created a list of 12 major hypotheses that we want to answer using the material that we are allowed, right? So the hypotheses that I'm mostly concerned about are hypotheses 1 and 2, maybe 3, yeah. So for example, the first one is, how does the sample compare with observations from a spacecraft, right? So we're going to compare the minerals, the thermal spectra, and see how it looks with other meteorites, and how does it compare? Because the analysis that I show you are from spectral analysis that we did while orbiting the asteroid, but we don't know how accurate they are, right? So that's one of the main hypotheses. We're also going to compare against Ryugu or Hayabusa2. This is a big one, how the chemicals of life, are present, what their composition is. So this is probably the most exciting question that we will be answering in terms of astrobiology. But of course, doing the assessment of the entire molecular distribution will also be important. So looking at all those different families that I showed you before. And and also, how Bennu can answer certain questions about the origins of the solar system. So, and then I guess the least exciting questions, but also really important, are related to the contamination assessment. Because if we want to use OSIRIS-REx as a model for future missions, we need to assess how, for example, the landing impact the contamination of the entire sample. So, so we recently developed an instrument. We built an instrument in the lab that focuses on the analysis of highly hypervolatile species that includes carbon monoxide, carbon dioxide, and so we're also going to be looking at that, those components, and yeah, so everything is tabulated, and those are the questions that I'm mainly involved with. So.
Yeah.
All right. Well, I'm gonna. Yeah. All right. Cool. Let's go online.
Yeah. Let's do like.
If we have more time.
Yeah, okay.
Of course, yeah.
Okay, another question online. Does the place of origin of asteroid affect its composition?
If the what?
The place of origin of an asteroid affect its composition.
Absolutely. So that's one of the hypotheses. That will tell us about the origins of the solar system, right? So we want to know if the asteroid migrated, how far, how close to the sun, if it was in its own orbit, like since it formed, and how that migration, that movement through the solar system affected its composition. And that's when the computational people can help, right? Because, again, we know what it's now, but we don't know how Bennu was 2 billion years ago. So what we see now may not be representative of the primordial solar system, and therefore we need to mimic that, right? It's a huge amount of work there, right? Just in that question, it's like you could spend years trying to understand what you see now versus what was before, right? So, and that, I don't think that, actually, this is a funny story because when I first got to Goddard, this is 11 years ago or something, and I didn't know about OSIRIS-REx. And I was talking with Jason, which is the science PI, and he's like, oh yeah, so we are involved in OSIRIS-REx, and we're gonna do this work and the analysis. And I was like, okay, but what we see now, how does it compare to what is before? It's like, I don't know. I was like, but nobody asked you? How do you get funded for this? Like, he was like, but that's something that we want to understand. That's something that we want to answer, right? And that's why we're bringing us a very pristine, a very biologically clean sample so that we can assess the inventory today and we can later. It's like I was talking with someone before that it's like imagine this drop of ink in water, right? And it spreads through the water. So we know what's at the bottom, how did it spread, but we don't know how it was at the beginning, right? And that's what we want to know. So, yeah, thank you for that analogy.
And the last question we have online right now is, how would the distribution of organic material collected from asteroids vary with collection depth?
Oh, I mean, that we don't know, right? So, yeah, well, that's something that hopefully we can see, but I don't know if we'll be able to assess surface versus in depth. Maybe by looking at different spots in the container, we'll be able to do that, right? So hopefully stuff that is in the touching pads are more surface, because these are Velcro. We didn't know if we were gonna be able to collect anything initially. We thought it was just like a dust flat surface, right? Hard, just dusty place. So we put Velcro, they put Velcro pads. And those maybe more surface, there will be also inclusions in the, right? So those are exciting. But then maybe the ones that are inside the filter will be more representative of the depth thing.
And we have one more question from our co-director, Francis Rivera Hernandez. How will the work on OSIRIS-REx help inform future analyses with Artemis on the moon?
Yeah, that's this hypothesis, no? What are the systems that we have to implement to appropriately keep the sample not contaminated from the Earth, right? I mean, we actually are involved on Artemis, trying to assess. For example, if you think about this, okay, we bring the material to the Earth, and you want to keep it safe. What container are you going to be using? Are you going to be using a plastic container, glass container, metal container, right? So you need to assess all of that. Are you using Teflon caps? Are you using what material is involved in the whole thing, in the clean room? So all of these details, questions are truly important for the safety of the sample. And OSIRIS-REx is going to inform a lot of that. So we think that at least 2% of the entire material will be saved under vacuum in a sealed container in a freezer. So, but then that brings major complications in terms of like, is it going to be glass? Like, are you going to like melt the glass, or are you going to be just cap it with Teflon, or and all those things are something that we continually keep monitoring. So.
All right. Well, thank you, Dr. Aponte, for this incredible talk, and thank you everyone for coming. And so I just want to remind everyone that we still have tons of food in the back, so please take some, and please give some to your friends who might not have eaten today. And also invite your friends to the social at 4 o'clock. Make sure you're there as well, and get ready for a fun astrobiology-themed event on the Mosey patio at 4 p.m. All right. Thank you all very much.
You had a really good race discussion.
Okay, that's a good number. Text three. It's nice to have that extra. Have a meeting. Recording will be there, so we can do Well, yeah. Is it? I've never looked at like the old videos when I video and actually
Yeah, no, fair enough. I was actually going to offer
Yes, it's my office.
Okay, I can take it.
Oh, okay. Or we should wait for the card, yeah.
Okay, I'm going to run up. I will be there. And then where can I leave it for everybody?
I wasn't last year, but if you got a bit of gummage. I know it's Boston this year.
Oh, nice.
Go all out. Well, whatever's not easy now, dessert.
No, totally.
Yeah, please have it now.
Yeah, absolutely. Yeah, have a as long as get
I have the membership, but I refuse to pay for stuff.
I know that you were in East Africa trying to call it. I have to tell me how.
That's
Yeah, the whole network.
I'll put it in there. I was showing her the.
Yeah, I don't want to
figure
you guys can give it all. Yeah, right now.
Yeah, so yeah, we were talking about.
And then I can just take it. This should probably be in the room, but like, nothing's gonna get back. Yeah, that sounds about
right. I understand, we don't just have like cash lying around and like 5-digit figures or something.
Right. She was like, I would be leaving.
$6,000 for your fine.
I'm trying to get you hooked up.
And you guys can you can tell them that I'll take them there for her, but that you want to come to the meeting, I'll take them for her.
I mean, it's a problem with everybody chemistry. I mean, you're right. We know the organizers back and post it. I for the last time. You don't even know.
Yeah, post it.
Post it today.
So great, yay. I'm gonna go.
And they want to jump into biosignatures directly.
Yeah, I don't know. We had some stuff. I think congratulations to you. I think pretty good. Amazingly coordinated. Like, what great timing. Oh, I'm so certified. Tell him I'll get him for his Like, we should probably
verify that he didn't have any errors in it. I made a Google Translate. I did the flyer, and then I made a Facebook-based post that I sent him the content. I was like, translate for me. I don't trust Google Translate. I don't know if I trust Google Translate either, you know? So, it posted, and no one was like, this is incorrect automation. It's automation shares from I'll take that.
I thought I was going to be a little nervous about that.
They did on the video.
And then we're there, and I'm like, oh, they're just like, no, it's really cool.
It was a reason I put it as 128.
Yeah.
You know, text 128. I walk in and I'm like, he's gonna be 228. It's not like a 7-hour room. It was like someone's laugh. Like, it's and I'm like, I hear it. I'm like,
I've got air. I'll wait. Yeah. I was like, coming out maybe like 3 feet.
whoop, and that's all gone.
And she points over my head and I look up. All I can see is the sun and I'm like 5 pieces left when I arrived.
And I arrived like 5 minutes
All
in.
right. I get up and then as I'm getting out, well, I didn't realize it was only here. I'm like on my way out. I didn't expect to see something. I was like, all right. Well,
I was like, oh, it looks small. I thought it was like mini muffin. $6 mini muffin. Okay.
Yeah.
Oh, wait. Yeah, thanks for the money. Alright, let's see how much we left.
You like organic chemistry? Oh, okay. That's cool. Yeah, so do I.
I'm so glad you guys enjoyed
If you want to start,
I didn't even ask.
I looked at it and I walked back and forth. Nobody was there. Yeah.
I
mean, if anything, it'll be because we use regular flow cell, you know?
no. Of course,
Yeah, yeah.
like funding.
Yeah, so we request the sample twice a year. And part of the panel.
Yeah, it's like 80 degrees. And they haven't been calling that much either, right? No, I'm one time. Is this new?
It is new. Is this new?
What new?
Probably got 2 bags.
Big day for both of us. How was Greenland?
Good.
Yeah, I loved your photo.
Impregnated in
I know a lot of the other programs have been better.
I don't.
What have you been up to? You guys went to the beach?
Yeah, we went to the beach.
Yeah.
I'll just send you the.
Yeah, no, could not stop it. They were like funny.
No, because I'm flying at 7.
Yeah.
Yeah, Seattle, we do work.
Yeah, no, but I just kept on like,
Yeah, I know him.
Yeah, I worked.
Well, he doesn't know me, but I know him.
Yeah, I was like, do you guys know each other?
Yeah, he works on your.
Yeah, I was
I really want to.
yeah. I was
So I set up.
So I'm going to underwater supporter for myself. Thank you so much. It was great. You're going down there what do you part of? It's like I've only been a part of this.
Yeah, we really excited how that's characteristics.
I'm excited to like show some videos and stuff when we finally go out there. Even just to saw like all the
features change
Yeah,
of course.
no, exactly.
That's why
I mean, we saw an eagle, right? We didn't get a video of it because it was so.
I was about to go to lunch. Alright, I gotta run. Are you heading down?
Oh, right. How are you?
So we're gonna take. I'll see you around.
Absolutely. Hello, how are you?
So nice to finally meet Chris.
Yeah, I think Julian and
Mando, Christy também estava enferma. Sí, no vino al lado. Ayer sí vino, ¿no?
Hey, engineers would have been done in like 5 minutes. Who would have gotten up to like half time? It's horrible. I'm like, coffee's not gonna be good afterwards anyway.
Do you have your card?
that's my truck.
Okay.
We might be able to get enough space. Yeah, if some days feel something.
Mucha gente que
At this point, I've done it like.
Mira how está like going around.
I'm not gonna make it.
So you're in
Yo, enfermé la semana pasada, pero no era como. I was like,
We'll pass my
I think I'm good. Solamente usé mascarilla en el.
Hopefully it'll improve after I graduate.
No.
I mean, I just found out.
No, y además lo publicaron súper tarde.
We'll walk down there. So, Luna's not coming.
I saw your tweet about it. Story. I could, you know.
You
were like, who wants to join me for the 3 apartments I'm looking into are all near a Starbucks on my way to work. I'm like, it's not great.
No,
It's not great for my
todos ellos. Siempre me dicen, yo como, ¿por qué no les hablan? Háblenles,
Exactly where the stores we have to if you're buying a $5 drink and you spend $200,
We don't have to get,
it's the same way that you spend $60 or $5. If you want to use it for a drink for 200 stores, just get like all the espressos, all the toppings, $300. Or save on 400 stores and you can buy one of those like nice coffee mugs. It's just 400 stars and that. Those cost like $20, $25. So I've gotten like an iced coffee mug and my coffee as well. So I know that it's nice. You gotta make it worth it, you know? It can't be just spending when it stars and
Willy-nilly. Yeah, I got for 100 stars yesterday, just like an iced tea.
oh, Dr. Rojas, amazing. Es nuevo. Los dijo como por 2 semanas, vengan a la clase antes del viernes de la prueba. Vengan. Pero sin las moléculas. Es como básicamente el formato de la
Alright, so
I forgot about the
Hay gente que viene a clase y no sabe las cosas. Hoy día estaba en Office Hours y una de las cosas que va a estar en la prueba son el nuevo diagram y un chico, el profe le dijo ya, alguien venga a dibujar el NO y el CO.
just get double next time. I just got, I was like, you know,
Para CO. Y yo iba a ir y dije como no, tú no, tú. Y
it's really important to practice rhythm.
no sabía cómo informarme.
Remaining cuts for